The overall goal of this procedure is to measure the relative cell cycle phase and cell size of tissues with cell populations genetically manipulated and labeled with GFP or RFP in Drosophila. First, set up genetic crosses to generate progeny containing cells labeled with the desired fluorescent proteins and with the desired genetic manipulations in a specific tissue or population of cells under study. Cleanly isolate the desired tissue from the progeny of the proper developmental stage. Then dissociate the tissue to a single-cell suspension and stain with a viable DNA dye to measure DNA content. Analyze the stained dissociated samples by flow cytometry. Ultimately, the in vivo results measure changes in cell size and cell cycle phase caused by the genetic manipulations under study.
The main advantage of this technique over the existing method of flow cytometry on live Drosophila tissues using ultraviolet dyes is that the DyeCycle violet dye is compatible with less expensive benchtop cytometers containing a violet 405 laser, which can be purchased on a small budget and maintained on a single lab scale. This method can help answer key questions in the cell cycle field by providing information on cell cycle phase and cell size that is more detailed than just measuring overall tissue size and proliferation rate. Demonstrating the procedure will be Kerry, Flegel, and Dan Sun, graduate students from my laboratory, and Olga, Grushko, our technician.
Collect mature larvae at the third larval instar at about 110 to 120 hours after egg laying for the larval dissections. Accurate pupal staging is essential for studies during metamorphosis. For this, harvest white prepupa at the